The overall goal of this procedure is to create a liquid elastomer device with a custom geometry using 3D printed mold pieces. This is accomplished by first designing the desired mold geometry in CAD software and 3D printing the mold pieces. The second step is to assemble the mold pieces into a full mold. Next, liquid elastomer is prepared and injected into the mold cavity. The final step is curing the liquid elastomer and removing the resultant device from the mold. Ultimately, this technique enables rapid creation of small-scale and low-cost medical devices in a resource-limited environment that would typically only be possible in an industry or larger-scale manufacturing setting.
The main advantage of this technique over traditional liquid injection molding is that it enables creation of elastomer devices in a resource-limited or academic setting. One may use this technique to create prototype medical devices for use in clinical studies to assess the feasibility of new medical technologies.
The ability to rapidly validate new medical devices at low cost and with a quick iteration cycle drastically reduces the barriers to device development and proof of concept realization. The method we've specifically developed is to embed electrical and optical sensors in an intravaginal probe device designed to scan human cervical tissue.
Generally, individuals new to this technique may initially struggle because of the design constraints involved in the use of 3D printed materials and multiple mold pieces to create a mold. We hope that our demonstration of this technique will help guide any future attempts to design a mold and carry out the necessary steps to yield any desired elastomeric device.
The first step is to use computer-aided design software to design a